And I'm going to introduce Dr. Peter Hansen here. So Dr. Peter Hansen is a distinguished professor and L.E. Rad Larson Professor in the Department of Animal Sciences at the University of Florida, where he has worked since 1983. Working primarily with cows, his research focused on the physiology and genetics of early pregnancy, thermobiology, and development of methods to improve fertility, enhance assisted reproductive technologies, and develop genetic and physiological strategies to reduce the impact of heat stress. He's a fellow of the American Association for the Advancement of Science, American Dairy Science Association, and American Society for Animal Science, and a distinguished fellow of the Society for the Study of Reproduction. With that, Dr. Peter Hansen, it's a pleasure to have you here. Thank you so much for accepting the invitation.
Yeah, thank you, JR, and thanks for that welcome. It's a real privilege for me to be here. You guys, as a group, have made fantastic inroads into the situation in terms of reproductive performance of dairy cows. It's really amazing. It's great for me to be here in Middleton. I spent the second year of my marriage in an apartment just up the road. While I was a graduate student at the University of Wisconsin. Actually, none of this was here back then. It was 1983. So Middleton has changed a lot, but it's always, I love coming back to the University of Wisconsin and to Madison area. The title is a great one. I did not come up with the title, but when I saw it, I thought, hey, this will be a great chance for me to kind of speculate, or you guys might think fantasize, about where reproductive biology is going to go in the next 20 years. So I've really enjoyed preparing this talk. I'm not going to talk all that much about in vitro fertilization. This opening picture here I thought was an appropriate one, really shows the very first step in the in vitro fertilization process, the collection of oocytes from donor female. This is Jeremy Block from University of Wyoming, and my graduate student Tatiana Maya from University of Florida. So this was taken in January in Florida. So you can see Jeremy here, he's wearing a t-shirt. Tatiana is all dressed up because it's very cold for her. And Jeremy is obtaining oocytes. So this is kind of a standard protocol now for many registered dairy herds, a great way to increase the number of offspring from genetically valuable females. This picture is also kind of interesting. This is not a typical female. This is a gene-edited Jersey cow that was produced by a collagen through CRISPR-Cas9 editing of the prolactin receptor. So in vitro fertilization is kind of a platform in the future for some other reproductive technologies. So I thought this would be a good picture to start off with. Thinking about the future made me think about the past, made me think about why is there a Dairy Cattle Reproduction Council? And of course, I was around as a faculty member when the DCRC was founded back in 2025, and it says right here on the website that the purpose of the Dairy Cattle Reproduction Council is to create a national forum and organization devoted to improving fertility in dairy cattle. And then if you read a little bit further in the preliminary vision, it says the vision was to increase adoption of reproductive technologies to counteract the observed decrease in dairy cattle fertility. So when the DCRC was founded back in 2025, the US dairy industry, probably the global dairy industry, was in the middle of this fertility crisis because of the continual decline in dairy cow fertility that had occurred during the past 40 years. And I borrowed this slide a few years ago from Paul Fricke, which kind of shows the situation back then and the situation today. So shown here is days open in the purple line, and then cow conception rate in the red line. So you can see that days open increased continuously from probably the earliest times it was being recorded back in the 1950s. So when the Dairy Cattle Reproduction Council was formed in 2025, some progress had already been made in reducing this increase in days open that had occurred historically, but we were still in a situation where dairy cows were infertile. And of course, since that time, as I said at the very beginning, there's just been tremendous progress in reversing the historical decline in dairy cow fertility. And you can just look at cow conception rate from 2005, when it was about 33%, to 2020, when it was 47%, to see the result of the scientists, the farmers, the technical support staff associated with the Dairy Cattle Reproduction Council and other groups to reverse this problem. So where do we stand today? We've achieved the goals of the Dairy Cattle Reproduction Council. So where are we going to go in the future? I'm not going to have all the answers to that, but our future investments in understanding reproductive biology of the cow going to result in the same kind of dramatic positive results that we've seen since 2005? Or are the returns going to diminish because pregnancy rate is already so high as compared to how it was? Or, are there other technologies that we can develop that can yield significant improvement in the efficiency and sustainability of dairy production? So I think you know how I'm going to answer this. Yes, there are other technologies on the near horizon and on the far horizon that can cause further improvements in the sustainability of dairy cow production. And I do like history, so I do like studying the history of dairy science. We've had a series of, I would say, historical challenges that the dairy science community and the dairy industry have tackled in the 20th century and into the 21st century. Artificial insemination, right? Probably the first cow in the US to get artificially inseminated was in 1901, but it probably wasn't really until the 1950s or 1960s that the technology to make artificial insemination a mature technology that could be widely adopted in the industry was achieved. And when I was a grad student in the 1970s, a lot of those faculty who developed artificial insemination as a practical technique were still in the departments with not very much to do because they had already perfected artificial insemination and didn't really know what to do other than tinker with it to make further strides. Also, beginning in the 1960s, as people started learning about the endocrinology of reproduction, a big effort emerged to develop estrus synchronization protocols so that we could control when cows were being inseminated. And then that was replaced with the development of timed artificial insemination by people like Milo Wilbank, Richard Pursley, and others. And, that is now a mature technology, widely used in the country. I think the first in vitro fertilization was in the 1930s by Pincus at Yale. And that led to a lot of research to capitalize on that technology to improve fertility in humans and to improve genetic selection in livestock. I would say that technology is still emerging, but by the 1990s, in vitro fertilization, embryo culture, embryo transfer had developed into a mature commercial technology that's used throughout the world. And like I just mentioned, beginning in the 1980s, there was worldwide recognition of the fact that dairy cows were infertile. And by 2020, or even before that, methods had been developed to restore dairy cow fertility. So when I was kind of growing up as a faculty member in the animal science department, it was great to be a dairy cow reproductive physiologist because all the funding organizations recognized how important it was to be able to improve dairy cow fertility. So now we've done it. So, what's next? So I'm going to propose 3 things that kind of vary in the magnitude of their importance, but I'll just list them all as great historical challenges anyway. I think we can improve procedures for in vitro production of embryos, and I'll just talk just briefly about that because that was in the title of the talk that I was given. I think the biggest challenge that people are working on now is to revolutionize the procedure for producing embryos by not so much in vitro production of embryos, but development of gametes from stem cells. So I'll spend about half my time talking about that. And then the third area I want to talk about, not as important probably as development of gametes from stem cells, is the idea that we can program the calf by changing the environment of the embryo or the fetus that it's derived from, or even by programming the environment of the sperm cell or the egg that calf was arose from. So this is a concept called developmental programming, and we've known about it since the 1980s. But just here at this meeting, there's a poster by Gabby McKay, who's standing right there in the front, showing how changing the environment of the cow right after the uterine environment of the cow right after insemination can improve the milk yield of the calf that's produced from the conception that arose at insemination. So I think especially these second 2 topics have the potential to further enhance the sustainability of dairy production. So let me just talk about these. What about IVF? IVF was not really developed as a reproductive technology, but more as a genetic technology. So with artificial insemination, we can produce hundreds of thousands of offspring from a genetically elite bull, but until embryo transfer came around, we could produce 1 offspring from a genetically elite cow. And then superovulation, where you increase the number of eggs released at ovulation, increase that number somewhat, but with the development of in vitro fertilization, we can further enhance the number of embryos and therefore the number of offspring produced by on the female side. That wasn't really so important until genomics came around. So even though we could produce lots of embryos from the best female, we didn't really know who the best female was. But once genomics came on board, the reliability of genetic estimates on the female side were just about the same as on the male side. So there was a lot more impetus to derive embryos from those genetically elite females. So I've always argued embryo technology should also improve fertility, because there's no loss of a potential pregnancy from failure of ovulation, from a defective oocyte, from the death of an embryo in the first 7 days of life. The embryo is placed into the cow, usually at day 7 after ovulation. Usually only healthy-looking embryos are placed in the reproductive tract. So we should have higher pregnancy rate with embryo transfer than we do with artificial insemination. But I'll show you some data in a minute. Except for situations when cow fertility is low, like during heat stress, the pregnancy rates to embryo transfer are really no different than due to artificial insemination. And then the last thing, I'm not going to talk about this now, I'll talk about it later. Since the environment of the embryo affects the phenotype of the calf, we can manipulate the environment of the embryo when it's in the culture dish to make a calf that produces more milk. So let me just talk about some of these things. So this is kind of the standard procedure for producing embryos by in vitro fertilization. Oocytes are recovered by transvaginal ultrasound-guided follicular aspiration, or oocyte pickup, OPU. So the ovaries are visualized with the ultrasound, then a needle pierces the vagina, pierces the follicle, and the oocytes are aspirated from the follicle. Holsteins are probably the worst animal to do this in terms of the number of follicles that can be recovered in a individual session. You know, we work with Brahman cows. You can get 50 eggs easy from a Brahman cow. Many Holsteins, you can get like 5. So oftentimes cows are treated with follicle stimulating hormone to increase the number of growing follicles on the ovary so that you can increase the number of eggs or oocytes recovered from the cow, which of course increases the cost. So then those oocytes are matured in the lab, right? They complete meiosis 1. And then at the same time, sperm are prepared. The sperm are capacitated in the lab. They're treated with heparin to make them capable of fertilizing the egg. Then the sperm and the egg are mixed together for somewhere between 6 hours, 18 hours. And then the embryos are washed to get rid of the sperm. And then they're placed in culture for about 7 days. And depending upon the culture system, somewhere between 20 and 40% of these oocytes become blastocysts, the stage in embryonic development when embryos are usually transferred. And then they can be transferred into a recipient cow and get a live calf. So how many embryos can you get? I'm sure people will argue with me about this number, and probably the companies that produce embryos for a living will say, no, we get higher. But if you figure roughly you get 5 transferable embryos per procedure, you can do IVF, or OPU, I should say, theoretically twice a week, but more realistically, people do it every other week. So theoretically, you could do OPU on an individual cow 26 times. So that would mean 130 transferable embryos per donor per year. You can do OPU on pregnant cows until they're about 3 to 4 months pregnant. You can do it on prepubertal animals. So as compared to superovulation and as compared to artificial insemination, you get a lot more embryos than would otherwise be the case. And this is a platform for other technologies, right? People are taking this basic model and building other technologies to run alongside in vitro fertilization. So at the beginning of this talk, I showed you a picture of a gene-edited jersey. We've been working with CMAX and Acelagen to produce animals with a gene edit that make them more resistant to heat stress. So what does that involve? That involves, you can do this many different ways, but one way is take oocytes collected by oocyte pickup, fertilize the oocytes, and then inject a series of enzymes and RNA molecules that cut the DNA and edit the DNA to make a change in the genetic code of the embryo, then transfer those embryos. And in a certain percentage of cases, the embryos will have been successfully gene edited. I mean, this can be as high as 75, 80%. So here's a gene edited Angus and a gene edited Jersey that have a modification in the prolactin receptor that make them produce really short hair and are more thermal tolerant. And there's a couple, here's a gene edited Jersey with this so-called slick mutation because she's got very short hair. Prolactin regulates hair growth, and this cow responds to prolactin differently than most cows, so she's got very short hair. Here's one of her calves that is also gene edited. And, you know, this sounds kind of like science fiction, but it is now legal in the United States to sell milk from gene edited cows that have the slick mutation, and it's legal to sell beef from gene edited cattle that have the slick mutation in the prolactin receptor. So gene editing is now a commercial reality. How useful is gene editing? I think if as geneticists discover mutations, single nucleotide mutations in genes that have a major effect on phenotype, like this gene does, then it's practical to use gene editing to introduce that gene into a population of cattle. So, you know, that's a reality now. I said before, we should get higher pregnancy rate with embryo transfer than we do for artificial insemination. So for this slide, I just took some data from the literature, so that mostly just from one experiment. What percentage of beef heifers artificially inseminated were pregnant at day 7 of pregnancy? What percentage of dairy heifers were pregnant at day 7? What percentage of lactating beef or dairy cows were pregnant at day 7? So that varies from 50% to almost 90%. But when you put a fresh embryo into a cow, an embryo that hasn't been frozen, I'm exaggerating a little bit, 100% of those recipients are pregnant, at least for a while. So you would expect using embryo transfer would increase the pregnancy rate as compared to artificial insemination. No ovulation failure, no early embryonic mortality, no fertilization failure. And for situations like heat stress, or when using repeat breeder females that have been inseminated 4 or more times and not getting pregnant, it is true that pregnancy rate to embryo transfer is greater than the pregnancy rate to artificial insemination. So here's the result of 6 studies comparing cows in the summer, inseminated, getting an embryo. You can see even when you get a high pregnancy rate to AI, still get a higher pregnancy rate to ET. So that's encouraging. Problem is, takes a specialized person to transfer an embryo. they usually charge $50, $75, $100 to transfer the embryo, which really makes the procedure not very cost effective. If we exclude situations like heat stress, repeat breeder cows, there is no difference in pregnancy rate between AI and ET. Here's a study from Roberto Sartori and Milo Wilbank, where, timed AI, 31% pregnant, timed ET, 34%. Another study in Brazil, 36% versus 33%. So there are problems. There's problems in how we make embryos. The embryo produced in the lab is not identical biochemically to the embryo that develops in the cow. And I would argue the procedures for embryo transfer are still not optimized. Basically, we use the same procedure we use for artificial insemination, except we give the cow an epidural. And I think, there's still a lot of optimization that can be done to make embryo transfer a fertility enhancing tool rather than just a genetic tool. Even though genetic tool, that's pretty good. The other thing, though, the improvement in ability to select females caused by embryo transfer still makes embryo transfer a pretty poor cousin to artificial insemination. I know many of you know this bull, Elevation. He's one of the foundational bulls of the Holstein breed. He not only was a good bull genetically, he had a very large ejaculate volume, so they were able to produce lots of straws from Elevation at select sires. In his life, over 500,000 straws of semen were sold from him. Supposedly, there's over 8 million descendants of Elevation today. So compared to that kind of impact, and there's advantages and disadvantages of that kind of impact, getting 130 transferable embryos per year from a genetically elite female still doesn't really equal the improvement that can happen on the male side, the genetic improvement. And that brings me to the second topic. One of the reasons that embryo transfer doesn't compare very favorably to artificial insemination, right? The bull probably produces about 5,000 sperm every heartbeat. Pretty good. The female, ovulates one follicle every 21 days. And she has other follicles growing on the ovary, but still the number is very limited compared to the bull side. There's a lot of interest in producing oocytes, eggs, and producing sperm, for that matter, in the lab from stem cells. And that's already been achieved in the mouse. And if we can do that in cattle, then we can produce as many oocytes from the best Holstein cow in the breed as we can from genetically elite bulls. So I don't want to bore you with too much developmental biology, but where do sperm and eggs come from? They come from bulls and females. They come from gonads, they come from testes and ovaries. But during embryological life, a specific type of stem cell called the primordial germ cell migrates into the developing gonad and differentiates into what's going to become either sperm cells in the male or oocytes in the female. So this just shows, here's a blastocyst stage embryo. The outer cells are going to become placenta. The inner cells are going to become the fetus. later in development, a few of the cells in this inner cluster of cells, the cells that are going to become the fetus, they become primordial germ cells. Primordial, the beginning, germ cells, either eggs or sperm. And then these cells migrate through the egg yolk, through the mesentery of the gut, and into the developing gonad. You can actually tag these cells with fluorescent dyes in the mouse and see them migrating through the connective tissue of the gut into the developing gonad. And once they enter the gonad, they differentiate into either oocytes or the sperm cell lineage. Well, people are starting to isolate these cells in the lab, and they can differentiate those cells in the lab into the precursors of oocytes and into the precursors of spermatogonia. In the mouse, they've been able to take these primordial germ cells, differentiate them into oocytes and sperm, achieve fertilization in the lab, and produce live offspring. In other species, to my knowledge, and I checked this before starting this talk, they haven't gotten quite that far, but they have made progress in taking these primordial germ cell-like cells and converting them into eggs and sperm. Now, where do you get primordial germ cells from? Well, you could get them from an early embryo. We've now learned how to take cells from the embryo and culture them in the dish while maintaining their ability to turn into different cell types. So we call those embryonic stem cells. So, I have those sitting in my lab. They're very easy to make. You can convert, not in the cow, but in other species, you can convert embryonic stem cells into cells that look like primordial germ cells. That's why that's called primordial germ cell-like cell. And then those primordial germ cells can be turned into eggs and sperm. There's one other thing you see here in this slide. Induced pluripotent stem cells. You can take somatic cells, skin cells, muscle cells, mammary cells, genetically modify them to act like embryonic stem cells and convert those into primordial germ cells and convert those into sperm and eggs in the mouse. So you could take, cell off your finger, make oocytes. So like I say, this has been done in the mouse. Both eggs and sperm have been made. I won't go through that. It involves culturing the cells in a whole bunch of different growth factors to kind of mimic the environment that the cells are living in when they're in the developing embryo. And this is a summary of the progress that's been made in developing offspring from oocytes and sperm that have been generated in vitro. So that's been successfully done in the mouse, in the human, and in the monkey. People have made progress at developing oocyte-like cells or sperm-like cells, but to my knowledge, they've not yet produced live offspring. I bet, actually, many of the breeding companies in the United States are trying to develop this technology because it would greatly change their competitive advantage in producing genetically modified animals. Pablo Ross, who is a scientist, used to be at University of California, Davis. He's now at ST Genetics. he published this paper back in 2019 saying that once we develop all these technologies, once we can produce embryonic stem cells from an embryo, well, we can do that now. Once we can produce primordial germ cells from embryonic stem cells, once we can produce eggs and sperm from the primordial germ cells, we can do our genetic selection in the lab. So the idea would be you take a genetically superior bull, genetically superior cow, genetic recombination is going to take place. The offspring are not all going to be the same. So you grow up hundreds of embryos from that mating, make embryonic stem cells from the embryos, do genomics on the embryonic stem cells to see which embryo has the highest net merit or whatever trait you're interested in, and then make gametes from those embryonic stem cells and produce more embryos and repeat the process again. So, if we can get this to work, I'm not saying we will, but I think there's a good chance we will. Then things that happen right now in vivo over a period of years could happen in the lab in a period of months. We could get several rounds of embryo production, genetic selection, more matings in the lab as we do in vivo. So that would be transformative. Good and bad, but generally good. So, here's where we are now with embryo technologies. We can take eggs from cows, multiple eggs, mature those in vitro, fertilize them, produce embryos, transfer the embryos to recipients. We can take somatic cells like a mammary cell or a skin cell. We can do cloning. We can take a cell from this animal, clone it by replacing the nucleus of this oocyte with the nucleus from the somatic cell. That's kind of standard technology now. So what could we do with embryonic stem cells and primordial germ cells? Well, we could take this embryo, make an embryonic stem cell from it, make a primordial germ cell from it, and then either make oocytes from it or sperm cells from it, and use those cells for in vitro fertilization. People at Cornell have developed techniques in the cow to make induced pluripotent stem cells. So these are cells derived from tissues like mammary gland, skin, muscle, liver, that have been tricked into thinking they're an embryonic stem cell. And those cells could be used to make primordial germ cells. So if these techniques come about, it really is going to change the way people think about genetic selection. How much time do I have left? 2 hours? 20 minutes? 15 or so? No, I will finish. All right, let me, I always put in too many slides, I'm sorry. So the third thing I want to talk about is programming development. This will be much easier to achieve than the last thing I talked about. When I was an undergrad at University of Illinois, I think the first thing I learned in Animal Science 101 was that an animal's phenotype is a sum of its genotype and its environment. So how much milk a cow produces depends on what genes she inherited, how much feed she gets, whether she was sick, whether it's hot, whether her herd mates are keeping her from the feed bunk, whatever. And we've made tremendous progress as people involved in animal production in finding ways to identify genetically superior females and males and propagate them. You just look at the improvement in milk yield over the years, to see how successful animal production specialists are in improving the genetics of animals used for food production. And we've made tremendous progress in optimizing the environment that those animals are raised in by providing the best nutrition, the best housing, the optimal photoperiod, treating disease with pharmaceuticals to optimize phenotype. But we usually do that after the animals are born. We don't really think too much about what is happening to those animals when they're embryos or when they're fetuses, or even when the germ cells that are being produced that will give rise to them are being formed. But what we now know is the environment of the mother that the early embryo is in can affect the postnatal phenotype of that embryo. The environment of the fetus can affect what kind of calf it becomes. A lot of work here at University of Wisconsin on effects of heat stress on the fetus. And I have to say, this kind of like boggles my mind too. The environment of the cow producing the oocyte can affect the phenotype that is formed at the end of gestation. And the environment of the bull. Arslan Tariq, I'm going to show some of his data here in a second, has data that how a bull develops affects his sperm in a way that affects the embryo. So we now have to think about environment, not just as something that we have to control after an animal's born, but before it's born. Let me just give you a few quick examples of this phenomenon. So when we produce embryos in vitro, we put them in an artificial medium. Among other things, they don't have any choline in there, the methyl donor nutrient. But in the uterus, choline is present at a millimolar concentration. So the embryo is really deficient in choline. So we did an experiment where we cultured embryos either in our normal medium or our choline medium, transferred the Brahman embryos, very bad dairy animals, to Angus or Brangus recipients, and then monitored the performance of the calves. And I think this is the best picture I've ever gotten from my research. You know, all these little Brangus recipients with these very hungry Brahman calves. We were weighing the calves, we let the calves go, they ran to their mothers, and my student Eliab took this picture. The calves that were derived from embryos that got choline in the culture medium were heavier at weaning than the calves derived from embryos without choline, right? So something that happened 500 days after that embryo was formed affects the weaning weight of the calf. We've done this experiment 3 times, and each time the choline calves weigh more than the calves without choline. That goes all the way through slaughter. So almost 3 years after these embryos were produced, the calves derived from choline-treated embryos had a higher slaughter weight, higher hot carcass weight than the vehicle-treated calves. Arslan Tariq, I think he's poster number 1. I didn't know he was coming to this meeting, so I'll talk nicer about these data. He looked at the effects of overconditioning bulls on the embryos that were derived from in vitro fertilization using the semen of those bulls. And you can see more details of that in his poster. But what he found was if you just look at the sperm, they look normal. I mean, sperm count's normal, motility's normal, fertilization rate is normal. But the embryos that are formed from bulls that were overnutritioned through a year of age produced embryos that a higher number of defects than control embryos. The last example I'll give, also another poster, I think poster number 2 from Gabby McKay. When I was an undergrad at University of Illinois, Phil Shuck taught us reproductive biology. He was a great scientist and a great teacher, really. But one of the things he told us was that seminal plasma was useless. I don't know what he's doing with that pig, but he said, you know, you can wash sperm, throw the seminal plasma away, you get normal fertilization. As far as he could tell, there was no role for seminal plasma in reproduction. So one of my colleagues at Florida, John Bromfield, when he was a grad student, removed the seminal vesicles from male mice, and the offspring from the matings of those male mice were different. There was an increased incidence of obesity, high blood pressure, etc. So John did an experiment that we got involved with that select sires played a crucial role in to evaluate the effects of infusing seminal plasma at the time of insemination on fertility of cows. So we did this project. That's Bo Harstein from Select Sires. That's seminal plasma. That's a lot of seminal plasma. We inseminated about 1,600 cows with a straw of semen and then a straw of PBS following the insemination, or a straw of semen and then a straw of 0.5 ml of seminal plasma. And we did not see a big effect on pregnancy rate or gestation length. Slightly bigger heifer birth weight for calves born using exsorted semen if they got seminal plasma. But for the most part, we didn't see much. So then Gabby, as part of her PhD thesis, followed these animals that were born in 2019 to see what was the milk yield of those animals that were derived from insemination with seminal plasma versus insemination with saline. And what she found, you can look at her poster, was that this is first lactation. We have about 169 cows. Those cows that were derived from a seminal plasma insemination produced about 5 pounds per day more milk than those cows produced without choline. So seminal plasma, which is full of biologically active molecules, change the environment of the uterus to change the way the embryo developed in a way that increased milk yield. And also the persistence of the animals in the herd was greater for those with seminal plasma than for those without seminal plasma. So, you know, if I was working for ABS or Alta or another animal breeding company, I would try to find out what signals can I put in semen extender that will program development of an embryo so that it becomes a heifer calf that produces more milk. I'm going to skip all this because JR is standing up. I'm going to skip all this. It's very fascinating though. So I will stop there. But I guess my take-home message is that, you know, we're not done. There's a lot of things that can be studied to enhance the performance of dairy cattle by manipulating reproduction. So thank you, JR. Thank you.
Questions? Questions? Hello, questions? You have time for one question. Anybody? Well, I can ask a question.
Yes.
So you said that the uterine environment and seems like to have a big impact on those. We are producing our next generation of animals mainly from heifers. What do you think is the impact of that we know that we're selecting genomically, but we are not probably providing as the best, I guess, environment for those animals, right? Maybe not the best nutrition in most cases. So
We did
should-
embryo transfer study where we looked at milk yield of heifer calves that were derived from embryo transfer where the recipient was the lactating cow, where the recipient was the heifer. And the cows that their mother was the lactating cow, they produced more milk when they were a cow than if their mother was a heifer. So I'm sure that is some kind of programming effect of being inside a heifer uterus. I don't know the specific cause, but yeah, no, there are environmental effects of the maternal environment on phenotype. Now, that's not gonna be inherited, right? That calf's daughter is not gonna have that,
But that's,
I think it's real.
all right. All right, we are out of time, so, thank you so much, everyone, and thanks, Dr. Hansen, for joining.